presentation. I'm very delighted to be here today and get to tell you about my research. So, I'm generally
We can't hear you.
Oh, I see. So, can you hear me now? So, I'm generally interested in the mechanisms by which brain circuit control goal-oriented behaviors such as feeding, and how these behaviors that are essential for survival are disrupted during eating disorders and drug addiction. So, today I'm going to show you about some initial studies to understand the function of a specific cell type and circuit in the context of feeding behavior, specifically about studies in the arcuate nucleus of the hypothalamus. So I will show you the new system we have developed to study feeding behavior in mice using the combination of photostimulation, genetic, and behavioral methods. So I want to start showing you pioneering experiments that have made significant progress to identify brain regions involved in specific behavior. For example, vision studies identify subregions of the hypothalamus in which circuits promote or suppress feeding. Those experiments strikingly suggested that circuits in the ventromedian hypothalamic nucleus are involved in satiety, whereas the lateral hypothalamic ones promote food intake. Consistent with this finding, electrical stimulation of the lateral hypothalamus trigger an increase in food intake. Moreover, traditional classical electrophysiology experiments show changes on the activity patterns of neurons in the hypothalamus during different behavioral states, such as hunger and satiety, as you can see in the raster plots for 4 different cell types. So, together, these classic methods identify the hypothalamus as a brain region controlling food intake. However, they're not extremely valuable for understanding circuit mechanisms because, as we know, neuronal circuits are composed of a diverse collection of cell types where each performs specific function and has a distinct set of synaptic connections. For example, remarkable work from the labs of Richard Paulnita and Tamaj Horvath in the arcuate nucleus of the hypothalamus show the two intermingled genetically specified cells known as AGRP, which express the agouti-related peptide, and POMC, proopiomelanocortin. They show that they positively and negatively influence feeding behavior. Classic pharmacology experiment has shown that both population of neurons might modulate downstream melanocortin receptors and influence food intake. So this is beautiful work, and I want to give massive credit to all these classic pharmacology experiments. However, I have something to say about that. So those experiments couldn't address whether the activity of those cells was sufficient to control feeding behavior. Moreover, they couldn't explain the relationship between neuronal activity and the magnitude and dynamics of feeding. And the pathway through which these cells function were unknown, basically to the limitation of those methods to genetically target identified cell types in different areas of the brain. So in order to address these questions, what we did, we developed a novel virus-based method that allows us to label cells for a specific neuronal stimulation. So briefly, what we have here, we developed a flip excision or flex switch, which allows for very tiny transgene expression, as in the absence of the transgene is inverted with respect to the promoter. So we took advantage of this system, and we tagged the light-activated cation channel, channelrhodopsin 2. I'm not going to mention so much as Lex already explained about it. So what we did is that we tagged channelrhodopsin fused to either a fluorophore, either M. cherry or TD tomato. Next, what we did is that we stereotaxically inject our 3 recombinant viral vector in an AGRP3 transgenic mouse, rendering those neurons photoexcitable. Next, we observed the expression, and what you see here, it's a coronal section of a brain slice of an AGRP3 channelrhodopsin positive mouse. And what you see is the M-cherry signal in the arcuate nucleus of the hypothalamus, which is the only location of AGRP cells in the brain. We also evoke photocurrents during whole cell voltage clamp and observe action potentials by applying brief light pulses in current clamp recording. So now we know that we can genetically target AGRP or POMC neurons We know that we can manipulate the activity of those cells in brain slices. But how to photostimulate those genetically identified cell types during a behavior, so in vivo? What we did is that we deliver light deep in the brain. We developed a system that allows us to deliver light deep in the brain. What we did is we implanted a guide cannula, and through that guide cannula, we implanted an optical fiber above the arcuate nucleus of the hypothalamus. For delivery of light pulses with millisecond precision, we split the output of a 200 milliwatt laser using the combination of beam splitters and Thorning mirrors. So each beam was controlled using acousto-optic modulators. By using two of this set of configuration, we were able to photostimulate simultaneously and independently 8 mice. So in order to then study whether AGRP neuron activation evoked food intake, we tested the mice for optically evoked food intake during the early life period, when mice are normally asleep and sated. So the protocol that we applied was similar to the activity pattern observed in brain slices in rats when you apply orexigenic peptides. So that protocol consisted of strains of 10 milliseconds action potential, so before, during, and after photostimulation. For the behavioral experiments, we placed the mice into feeding cages. As you can see here, in these cages, the mice have food pellets delivered ad libitum. They also have access to water without restraint. So this video is going to show you an example of an AGRP3 channel rhodopsin positive mouse that is being photostimulated for the first time. And as I told you before, this is during the early life period. So you can see here the time in minutes, and the red flashes are going to indicate when the animal grabs a pellet. You can see that the photostimulation is on, and again, in the early life period, she's asleep, not interested in food at all. But just pay attention about the time. Just within minutes of photostimulation of AGRP cells evoke voracious food intake. Moreover, the food intake was evoked selectively over drinking because, as I told you before, the animal has access to water without restraint. So this result shows for the first time the causal link between AGRP neuron activation and feeding behavior. Now, in order to understand how AGRP neuron, or to gain insight how this AGRP neuron activation evoked food intake, what we did is we analyzed the feeding response. First, we plot the number of pellets eaten by an AGRP3 channel rhodopsin positive mouse, and also by AGRP3 animal with not channel rhodopsin expression as a control. What you see here is the number of pellets eaten before, during, and after photostimulation, and we see the photostimulation of AGRP cells evoke voracious food intake within minutes. So, in addition, when we plot the magnitude of food intake and the latency to food consumption versus the number of AGRP neurons expressing channel rhodopsin, we found that it's proportional to the number of cells expressing channel rhodopsin. So also, the magnitude of food intake was similar to the amount consumed during refeeding after 24 hours of food deprivation. So these results displayed prove that a single group of neurons, at least 800 channel rhodopsin, 800 AGRP neurons, are able to evoke a complex behavior such as feeding. We next ask, is just the number of neurons that are activated, or is the rostrocaudal distribution or the position of those cells throughout the arcuate nucleus of the hypothalamus? So what we did is that we quantify that. So what you see here, it's an example of 6 representative mouse. The red dot shows you the distribution or the expression of channel rhodopsin expressing AGRP cells. What you see here are the bar graphs showing you the number of neurons in each rostrocaudal position. When we plot that, you can see a variety of distribution. However, when we plot the food intake for these 6 representative mouse, we found, with those 6 representative mice, we found that the food consumed was similar to mice with greater than 800 channel rhodopsin cells, which somehow indicates that the distribution of the cells, it's not what it counts, it's just the amount of cells that are activated. So pretty much by the same time that we were photostimulating AGRP neurons in, using optogenetics, Mike Crashes in Brad Lowe's lab at Harvard, now he's a tenure track investigator at the NIDDK, it's actually Leg's neighbor, was trying to evoke food intake by stimulating those neurons. But what he did is that he used the DREAD system. In this system, the G-coupled protein receptors are mutated, allowing a compound known as CNO to bind to those cells and activate them. So that was pretty much like a race between labs, but in the end, Mike and I were really, really glad because we both came out with very similar results. However, one of the many advantages of optogenetics is that allow us to change the activity patterns of those cells and challenge the system. So what we did next is we basically applied different stimulus frequency on those cells and studied the feeding response. So we applied 20, 10, and 2 hertz photostimulation protocol, and what we observed is it's frequency dependent. In addition to that, the evoked food consumption was consistent across subsequent stimulation trials, as you can see on the second trial of 20 hertz photostimulation. So what I've shown you so far is that we have developed this system that allows us to rapidly manipulate neuronal activity in the brain. But what about circuits? So we're interested about which of the pathways through which AGRP neurons regulate that food intake. So what we did next is that we analyzed downstream components of the circuit. As I told you during the introduction, POMC neurons are intermingled with the AGRP neurons in the arcuate nucleus of the hypothalamus. AGRP neurons inhibit POMC cells. Pharmacology experiments have shown that both cells influence feeding behavior by acting on downstream melanocortin receptors. So we also manipulate the melanocortin pathway. But first, we photostimulate for 24 hours POMC3, POMC cells in the POMC3 general dose in positive cells, and quantify the amount of food intake and body weight before and after photostimulation. And you can see the photostimulation was sufficient to reduce food intake and body weight. We next asked if this hypophagia was mediated by through the influence of the melanocortin pathway. In order to study that question, we used the agouti mice. In these mice, the melanocortin receptors are blocked by the constitutively expressed agouti protein. So the phenotype of this mouse is very similar to this big guy here. So they're obese, the coat color is yellow. So what we did is that we crossed our POMC3 mice with the A1 mice to generate POMC3 A1 mice. Then we photostimulated POMC cells on these cells, and surprise, the hypophagia was not observed anymore, which indicates that the melanocortin pathway is required for the POMC-mediated hypophagia. We next asked whether the melanocortin pathway was required for the acute AGRP-evoked food intake. What we did is that we photostimulate AGRP cells on the AGRP3 A1 mice, and we observed that the photostimulation evoked voracious food intake, and this amount of food consumed was similar in magnitude to the amount of food consumed when we photostimulate the AGRP3 mice, which is suggesting that the inhibition of the melanocortin pathway through the AGRP neuron activation is not a necessary condition for acute. And I made it clear that it's acute because we're just photostimulating for 1 hour, right? So we don't know what's going to happen after 24 hours stimulation. There are all these satiety cues that can kick in. So with this, I basically would like to summarize this part of my talk. I have shown you new methods, photostimulation, behavioral, and genetic techniques to study goal-oriented behaviors, to identify cell types in the circuit. I have shown you that AGRP neuron activity is sufficient to initiate and maintain food intake. And I show you that the melanocortin pathway is not required for the acute evoked food intake by AGRP stimulation. Now, I would like to show you a little bit more what we want to do on my lab at NIDA. So, we're taking advantage of the methods that I have developed in the Sternson lab for literally cracking the neuronal circuits underlying the rewarding nature of food intake and drug abuse. So what we want to do is we want to identify every cell type involved in that specific behavior. We want to understand the activity patterns of the neurons during behavior and understand how these cells function together through synaptic connection to regulate circuit function. So we're going to start with the lateral hypothalamus because, as I told you during the introduction, translesion, pharmacology, and electrical stimulation experiments is known that the lateral hypothalamus is somehow regulating food intake. I'm really excited about Garrett's talk because I'm pretty sure he's going to tell you more about the lateral hypothalamus and the hypothalamus in general. So it's also known that the lateral hypothalamus regulates other natural behaviors such as maintaining water consumption, body temperature, circadian rhythm. In addition to that, the lateral hypothalamus is densely connected to other areas important for food intake, such as the arcuate nucleus of the hypothalamus, and areas important for goal-oriented behaviors, such as the nucleus accumbens and the ventral tegmental area. Moreover, they genetically identify and distinct population in the lateral hypothalamus, making an ideal circuit to study the neuronal basis of behavior. For example, neurons expressing melanin concentrating hormone, MCH, neurons expressing hypocretins or orexin, and leptin receptor expressing neurons can be all target genetically for optogenetic experiments because mouse lines expressing CRE for those cells are available. And last but not least, we have developed the tools to target and manipulate hypothalamic cell types during behaviors. So what we're doing in my lab now is we're starting to manipulate these lateral hypothalamic cell types using fine spatial and temporal resolution optogenetic techniques. So we want to track all the axonal projection and manipulate those cell types using either channelrhodopsin, arcurodopsin, and study the behavioral outputs. So in addition to that, and in collaboration with my very good friend and colleague, Naji at Genelia Farm, we're trying to implement optic techniques. We're trying to image many neurons in the brain using two-photon fluorescent endoscopy. So the Yeka has spent at least 6 or 7 years developing new scopes and imaging techniques that allow us to image with high spatial and temporal resolution deep in the brain. So what she does is that she uses adaptive optics in two-photon fluorescent endoscopy, and through green lenses or gradient refracted index, we can image at least 6 millimeters deep. So Miriam Bukarski, she's a postdoctoral fellow in my lab. She's now training at Genelia together with the G postdocs. And what they sent me recently, they have been trying to characterize the system. So these images are taken from a brain slice of a GFP transgenic mouse. This is in vitro, but they're using a green lens. And what you see here, this is the image before the correction for aberration, before applying adaptive optics. And this is the image after they apply the correction. And as you can see, it's remarkable better, which is very promising for us because we're trying to image the axonal projections of the hypothalamic cell types. So that is very promising. And I don't know if people at the audience are working in the hippocampus or the cortex. As you know, those structures are beautifully laminated. When you're trying to record for those cells, you get a very high throughput. However, in the hypothalamus, because the geometry of the cell, it's really, really hard to record electrical activity. So now we're trying to compensate for that using imaging. And the other good thing is that recently they improved the GCAM. So now we're using GCAM 6 to observe the terminals with high resolution. So ultimately, because as I told you during the introduction, as the lateral hypothalamus is densely and highly connected to the nucleus accumbens in the ventral tegmental area, we would like to test the hypothesis that the lateral hypothalamus it doesn't function just as circuitry for food intake, but maybe it's involved in modulating other goal-directed behaviors such as drug abuse, mating, and water consumption. So, and with that, I would like to wrap up, and I hope that I have convinced you that previous studies have often treated the lateral hypothalamus as a homogeneous structure. So our goal is to identify and determine the functions of each component on that circuit. And I strongly believe that by understanding the mechanisms how these cells function, we will enable our ability to battle disorders such as obesity, addiction, diabetes, bulimia, and anorexia. With that, I would like to conclude my presentation and acknowledge my postdoctoral advisor, Scott Sternson at Genelia Farm, for his guidance throughout this incredibly exciting research, all my colleagues at Genelia Farm that contributed to my work, and my lab members at the NIDA for their dedication and incredibly patience while building a lab from scratch. And thank you all for your kind attention.